So, we are getting close to the end of this course. Today I want to talk about nitric oxide, and then we have 2 lectures left, I believe. I'll present a little bit the history and the discovery of actually, this is not working, of nitric oxide. How is it synthesized and regulated? Its paracrine effects, signaling, of course, intracellular signaling. How is it involved in regulation of vascular tone? And then basically going into some of the functions and some of the new discoveries new discoveries, they are no longer new. What functional roles nitric oxide actually has. But if we start from the very, very beginning, I'd like to ask, what molecule is this? Does this molecule resemble something? And of course, in the light of the fact that you already have the answer, it's a bit of ridiculous to try to discuss that, right? But this molecule here is nitroglycerin. And you know very well what nitroglycerin is used for, right? Nitroglycerin is used as an explosive, stabilized, it's used as dynamite, and of course that's what Alfred Nobel was very well known for, etc., etc. But even at the time that Alfred Nobel was alive, nitroglycerin was used for something else. And it was used to treat angina. Angina is this clinical symptom in which people feel like this oppression in the chest, something that basically is failing, that the heart actually aches. And this is known to be related with an impending heart attack. So obviously nitroglycerin is not only a potent explosive, but physiologically it does something. And this molecule, nitroglycerin, that has been used for now more than 100 years in even the treatment of angina, we knew it worked, but we did not know through which mechanisms. Angina basically characterized by this pain in the thoracic region, accentuated by situations of exertion, heavy meals, cold and smoking. And if you want, this picture here portrays everything, right? This businessman that basically has been eating at a restaurant with some company representatives or whatever. You obviously just basically going out in the cold, well, presumably also smoker. Well, this is the typical compounding or the association of risk factors that will basically treat or conduct or conduce to angina. And we know that angina pectoris, angina is this pain in the chest, is related to cardiac ischemia. And cardiac ischemia is actually meaning that the heart, the myocardial muscle, is poorly perfused with blood. What the picture here is showing is a picture of the vascular cast or the vascular beds that supply blood to all the cardiac cells. And what is it that you see here, basically it's a set of blood vessels, the coronary vessels, that they originate all in the aorta through a small hole, and basically the blood goes out into the entire cardiovascular, the entire myocardial muscle. So when something happens with this, what could happen with this? Well, usually the typical problem with angina in a situation of a heart attack is that one of these major vessels basically gets occluded and stops actually providing flow. If this major vessel here, this major artery, is occluded at this point, the downstream region, the region downstream of the blood vessel will not receive blood, therefore will not receive oxygen. What will happen to the cardiac tissue? It dies. It dies. And that means that less of the heart will have to actually perform as well. So basically part of the heart is no longer pumping and there's necrosis. But as I told you, organic nitrates, nitroglycerin and others have been used for treating angina since the beginning, the middle actually of the 19th century. the mechanisms were not understood. Alfred Nobel and many others afterwards actually took sublingual nitroglycerin, these small pills that you put under your tongue, basically that alleviate angina. How do you think they alleviate angina? Do you know how they alleviate angina? Nitric oxide that now we know is released from nitroglycerin is actually a vasodilator. So what we will be presenting in this lecture is this vasodilator role of nitric oxide. But this vasodilator role has a very interesting history. And the history goes back to the 1960s with a guy called Robert Furchgott. Robert Furchgott was doing some interesting experiments that basically led him to believe that the endothelium of blood vessels produces a vasodilator. Let me explain the experiments that Furchgott was doing, which I'm trying to illustrate here. First of all, Furchgott took blood vessels. You know what a blood vessel looks like, right? It's basically like a tube. And he took these blood vessels and prepared them in different ways. In a way, what he was doing is something very similar to what you are doing in the lab. Instead of using the cardiac strips, the strips of the heart that you take, right? He was using blood vessels. And then, how do you put a blood vessel in the system? How do you do it with the strips of the heart muscle? You cut a region, right? And you tie it on both ends. Well, how can you prepare in a similar way a blood vessel? There are two ways of doing it. One is the ring preparation. Now, imagine my arm is the blood vessel. If you cut it through the middle, do a cross section, you can do a ring, right? A ring, basically, that's going to be this very short tube, right? So this would be this is the bit of the ring. And you can prepare that how? Well, what you could do, right, is basically hook the hooks or a thread on both sides of this ring, stretch it, and then you hang it that way, right? So basically your ring, instead of being round, now becomes this, because you have tied it, you have stretched it, and then you can measure the force that this ring is producing. Why does a blood vessel ring produce force? That's an important question. Why does a blood vessel ring produce force? Why does a cardiac strip produce force? Because in the strip, in the cardiac strip, you have contractile muscle, right? Which kind of muscle? Cardiac muscle. Why does a blood vessel ring produce force?
The same reason, contract and baseband.
Which kind of muscle is included in the blood vessel wall?
Smooth muscle.
Smooth muscle. So you have another type of muscle, but you have a muscle. So you can do it with a ring preparation. But you can also do it in a helical strip. In a helical strip, instead of actually hanging this ring and stretching it, what you do is cut it. You take the blood vessel and then put the scissors and cut it, but instead of cutting it straight, you cut it in a sort of an helix way, so that at the end it will stretch as a fold, right? Do you see that mentally? So basically, you cut in a kind of spiral way, and then when you stretch it, it's going to be a flat piece, and then you can prepare it the same way. What Furchgott observed in the 60s is that if he prepared a blood vessel in these two ways, he obtained different responses. This is the experiment. The experiment always starts with a dose of noradrenaline, in this case, any norepinephrine, but it's noradrenaline, right? So you give a bolus, a concentration of norepinephrine. What does that do to the smooth muscle of the blood vessel wall? It constricts, right? Noradrenaline is a vasoconstrictor. Through which kind of receptor? We have studied this in the course as well.
Beta 2, GS.
No. vessel, vasoconstriction. Alpha adrenergic receptors. We have mentioned that beta adrenergic receptors are involved in vasodilation in some specific blood vascular beds, the muscle, for example, in the skeletal muscle. But in most of the body, the blood vessels constricting are actually due to alpha adrenergic receptors. But so through alpha adrenergic receptors, you get this noradrenaline response constriction. And then what they do is they do a dose response curve with a substance called carbachol. What is carbachol? Carbachol is an acetylcholine agonist. It's an acetylcholine agonist. So when you apply acetylcholine to these blood vessels, what do you see? A vasodilation. Do you see the vasodilation? As increasing doses of carbachol or acetylcholine, this basically ring in this case vasodilates. The interesting thing is that if you do this experiment on a helical strip, if you cut the blood vessel in a different way, as I told you, you don't get that response. As it so happens, when Furchgott obtained these results, he could not explain them, and he put them in a drawer in his office. He could not explain these results, so basically, well, time will tell. And essentially, time told after a while. The first impression that he was getting is, of course, that in this blood vessel, something actually makes the blood vessels to constrict, to dilate. But since he could not reproduce it in other ways, I mean, he just never forgot about it, but did not do much work. One thing more that he realized, though, is that the effect, the differential effect associated with a ring or a helical strip, was actually dependent on rubbing the blood vessel or not. Here is the experiment. If you do a ring, you get vasodilation, as I told you earlier. But then he realized that if instead of only taking this ring and hanging it and doing the experiment, he took the ring, rubbed it a bit, and then hanged it, the response disappeared. The same happened with the strip. I told you with the helical strip, he did not get vasodilation, right? But when he started doing it more carefully, he realized that if he was very careful at the time of cutting, he could get the vasodilation. But then if he rubbed it, the vasodilation disappeared. What that told him, well, he did other treatments, what that basically told him is that the part of this blood vessel that was important for this response was the endothelium. Dennis.
What's the W?
That this is a wash. The W means when you wash. So basically they clean up, they just remove the drugs and they put some fresh saline or buffer. So then of course you go back to the beginning. That's the wash. So are you with me? So basically the difference here seems to be in the endothelium. What is the endothelium? What is the endothelium? Peter?
May I take it in Swedish?
Yeah.
It's the layer, ett lager innanför och innanför som olika, eller endotheliumceller och sen olika specifika.
So if we now split up the blood vessel in different layers, the endothelium would be the one closest to the blood or farthest away from the blood? Closest to the blood.
And it will be exposed when he cuts it.
Exactly. So the endothelium is the very thin layer of cells that actually lines up the lumen of the blood vessel. And it turns out that this experiment is saying that this layer, which is very thin, is basically a unicellular cell layer, is very important for the responses of the actual blood vessel wall in terms of constricting or dilating. This is the main observation that Furchgott actually did. People kept working with this and studied this, and then they realized they created this sandwich proceeding which is a very smart procedure, in which if you take the smooth muscle layer of the blood vessel alone, and you apply acetylcholine, you get vasoconstriction. However, if you now, beside your layer of smooth muscle, take another blood vessel, this is another blood vessel, with the endothelium intact, and you apply it in contact with your smooth muscle layer, when you add acetylcholine, you can get vasodilation. So this experiment, the sandwich experiment, is proof that something in this layer is happening, something in this layer is released that actually triggers vasodilation. And this is what became called the EDRF, the endothelium-derived relaxing factor. Endothelium-derived relaxing factor. I think I do have it here. And EDRF, endothelium-derived relaxing factor. So what this is showing is that EDRF, this factor EDRF, diffuses from the endothelium into the smooth muscle and causes vasodilation. Nevertheless, we knew of the existence of the EDRF, we knew that it was produced by the endothelium, but we did not know some other things about it. And here comes a second person in the picture, and that's Ferit Murad. What he discovered is that the signaling associated with EDRF went through a molecule that actually it was cyclic GMP. What Murad realized, by that time they knew, when he was studying this, they knew that many signaling mechanisms, signaling pathways in cells actually were mediated by cyclic AMP. We have studied plenty of cyclic AMP mediated processes. You have done a lab measuring cyclic AMP after signaling, after stimulation with adrenaline. What he realized is that cyclic AMP did not change in a situation of vasodilation. The molecule that actually changed and was related with relaxation was actually cyclic GMP. So Ferit Murad is the one that realized that the signaling factor associated with EDRF was cyclic GMP. And that's another small bit into the puzzle. However, we still did not know what was EDRF. And the person that actually discovered this was Luis Ignarro. Ignarro realized that this EDRF actually, well, the main problem in discovering what EDRF is basically for the answer that we know today. We know that the endothelial-derived relaxing factor is actually nitric oxide. Nitric oxide is a gas. Nitric oxide has a very short half-life in vivo. So, of course, it was not an easy molecule to find. It was not an easy molecule to find. How did Ignarro found out about this? Is because actually nitric oxide changes the spectrum, the absorption spectrum of hemoglobin. So by analyzing spectral changes in the hemoglobin associated with the release of this EDRF, he characterized the molecule that was actually responsible for this, and that was the nitric oxide gas. EDRF released from the endothelium, but also nitric oxide. So he did experiments with both and realized, well, they do exactly the same thing. They shift the spectrum of the hemoglobin in exactly the same way. That told him that EDRF must be nitric oxide. Yes, Rebecca?
What do you mean with the shifts, the absorbance spectrum?
Yeah, I don't have the time to go into that, but basically when hemoglobin binds, the hemoglobin binds oxygen, there are some big shifts associated with the binding of oxygen. The same thing happens when hemoglobin binds carbon monoxide, for example. The same thing happens when hemoglobin binds nitric oxide. So there are some shifts, and this is what this is trying to represent. The absorption spectra at different wavelengths shifts because nitric oxide, when binding to hemoglobin, create some conformational changes that can be picked up in this way. Well, these three people together put together basically the key aspects of nitric oxide signaling. And that's the reason that they got the Nobel Prize quite a few years later, actually. This was actually an expected finding. There was a lot of people working with EDRF at the time, so of course it was well known that whoever found out these things basically would win the Nobel Prize, just essentially because nitric oxide has been demonstrated to be important in many different mechanisms. As always with Nobel Prizes, there can be discussions, and in that particular case there was a fourth person that also had contributed importantly, but the Nobel Foundation has always, that's the way it should be, 3 persons can get the Nobel Prize, so that fourth person was excluded, and I'm not even going to mention it in this lecture. But that's the way it is. Here it is. The mechanism is basically what I told you. You have the endothelial cell, this single layer of cells, under which you have the smooth muscle layer. The smooth muscle layer is actually a multi-cell layer, and what happens is that the endothelial cell releases nitric oxide. We will see the details later. It releases nitric oxide. This is shown in these green balls here. In the case, one of the mechanisms for releasing nitric oxide is a stimulation of the acetylcholine receptor. This is not the only mechanism. This is the mechanism that actually was used by Fulchgott to do the part of these studies, but this is not even the main physiological mechanism for activation of nitric oxide. Nitric oxide produced, released by diffusion into the smooth muscle, and what happens when it reaches the smooth muscle cells? Well, nitric oxide in contact or in the smooth muscle cells basically cause the breakdown of GTP into cyclic GMP, and this is actually the discovery of that. Cyclic GMP concentration increases, and that leads to a decrease in muscle contraction, vasodilation. All these small steps, we will see them in a second. Pay attention to the point that half-life of nitric oxide in the circulation is short. This is a gas, and it's a very simple gas that degrades quickly, 2 to 30 seconds. That's the half-life. Any questions so far? Rebecca?
Yeah, you talked a little bit fast, but what was it with the GTP and cyclic GMP?
Yeah, basically nitric oxide, when nitric oxide diffuses in the smooth muscle, it actually targets, and we will see the exact details, but it targets basically an enzyme here, we will give it the name in a second, to actually convert or break down GTP and form cyclic JMP. And cyclic JMP in this case would be the second messenger in the cell. Notice that nitric oxide is a gas. Do you expect a receptor for nitric oxide in the cell surface? No? Why not beta?
It should just pass.
It will diffuse through the plasma membrane.
It's so small.
Exactly, it's a very small molecule. It will diffuse through the plasma membrane, so that means that this enzyme here is a cytoplasmic enzyme that will actually break down GTP into cyclic JMP, and the cyclic JMP is going to be the second messenger in the smooth muscle cell. Let's look first at the synthesis of nitric oxide, which happens in the endothelial cell. And basically, this synthesis is catalyzed by an enzyme called nitric oxide synthase. It's a molecule that synthesizes nitric oxide. The substrate for this enzyme is L-arginine. L-arginine is an enzyme, is an amino acid, as you well know, right? So L-arginine, the amino acid L-arginine, is actually the substrate for the formation of nitric oxide by nitric oxide synthase. We know that there are 3 isozymes, 3 isoforms of nitric oxide synthase. NOS1, nitric oxide synthase 1, is also called the NNOS, or the neuronal NOS. this is a synthase that is constitutive. Constitutive means that it's an enzyme that is working permanently and it's producing nitric oxide. And this happens in the neurons. We know that nowadays that nitric oxide is also called a gasotransmitter. It's a neurotransmitter because it's a gas, it's called a gasotransmitter. Central nervous system. Nitric oxide synthase 1, the neuronal NOS, is actually a free cytoplasmic enzyme. And then we have NOS2. NOS2 is the cytokine-inducible NOS. This NOS, this nitric oxide synthase, is found in macrophages, it's found in the liver, in a smooth muscle, in the endothelium, in the heart as well. It's also a free cytoplasmic form. Finally, we have NOS3. NOS3 is the endothelial NOS. It's the one that we have been talking so far and the one that we will spend most of the time in. Endothelial NOS is actually a constitutive nitric oxide synthase found in endothelial cells, found in blood vessels. It's a membrane that is actually associated to the plasma membrane. It's in the cytoplasmic portion of the cell, but it's associated with the plasma membrane. How this synthesis occurs, or well, how is this then regulated in vivo? And now we are going to talk only about eNOS, which is the endothelial form of nitric oxide synthase. The major in vivo factor for triggering nitric oxide release is actually what we call shear stress. I mentioned shear stress in this course before, but essentially shear stress has to do with the friction that blood provides on the blood vessel as it circulates. And notice that the endothelium is, of course, is in contact with the blood. So it's through this shear stress that is the major physiological stimulus for the synthesis of nitric oxide in vivo. We also know that acetylcholine, through the muscarinic receptor, through one of its receptors, and that is actually a GPCR, G protein coupled receptor, can also stimulate nitric oxide release. And in a few slides, you will see the mechanism how that happens. Acetylcholine effects, if you use this to trigger, basically acetylcholine effects are related to an increase in intracellular calcium. This increase in intracellular calcium activates phospholipase C. Now we are in the endothelial cell. In the endothelial cell that releases nitric oxide. One of the mechanisms for nitric oxide release is shear stress. Another one is activation by acetylcholine. So this mechanism is the acetylcholine mechanism. Acetylcholine is the ligand, binds to its GPCR, a muscarinic receptor. And that actually, through the activation of your well-known and understood phospholipase C, triggers the breakdown of PIP2 into IP3 and diacylglycerol. Should I ask you which G protein? You know, GQ. And triggers the increase in intracellular calcium. And this is when this happens, you have the release of nitric oxide. When this nitric oxide is released, it's a small molecule, it can cross plasma membranes, both the endothelial cell, and it will diffuse into the smooth muscle. And there, it will, or it can bind to an enzyme called guanylyl cyclase, GC. Guanylyl cyclase is a soluble enzyme. It's present in the cytoplasm of the smooth muscle cell. And there, it will actually trigger the formation of cyclic AMP by degradation or by breakdown of GTP. When that happens, cyclic GMP increases, and that leads to myosin relaxation. So the net effect is that nitric oxide diffuses, you have a cyclic GMP responsive enzyme, and then you have a smooth muscle cell relaxation. So increase in nitric oxide, vasodilation. We will discuss this effect later in terms of the in vivo or its physiological significance. if we look at guanylyl cyclase activity, or if we look at guanylyl cyclase, which is actually the enzyme that forms, that will form cyclic JMP. So here you have a very simple sketch that illustrates the generator cell, that you know it's the endothelial cell, breaks down arginine into citrulline and nitric oxide. Nitric oxide diffuses out of the endothelial cell into the smooth muscle cell. And here you have the soluble guanylyl cyclase enzyme. In this soluble guanylyl cyclase enzyme, nitric oxide binds to the heme group, and there it basically forms cyclic GMP. The binding of nitric oxide is what makes this enzyme particularly active. So the catalysis, the formation of cyclic AJMP, when nitric oxide is bound to the heme group, is increased in 2 or 3 orders of magnitude. And remember, 2 or 3 orders of magnitude in this case would be 100 to 1,000 times more activity. So basically the picture here is that you have the soluble guanylyl cyclase that is actually breaking down GDP to cyclic GMP very slowly, but when nitric oxide binds, then this action is catalyzed much faster. Any questions up to now? Because now we are going to talk about the physiology of nitric oxide, and this is of course the basis of understanding that physiological effect. So physiological effects of nitric oxide, there are many functions of nitric oxide. It's part of the host defense cytotoxic mechanism, so it's actually involved in the immune system. It's also a mediator in the central nervous system. That's why we call it a gasotransmitter. And there it's associated with excitatory amino acids, glutamate and glycine. It has an effect on platelets, on coagulation. And that's basically because platelets can release nitric oxide as well. But the effect that we will be discussing for the rest of our lecture now is going to be the paracrine regulation of vascular tone. And if you remember, we have talked about paracrine mechanisms in this course before, and we defined them as well. What did we say was a paracrine mechanism?
It's when a cell stimulates another cell close to it.
Exactly. And that's exactly what we are talking about in the case of nitric oxide, right? Endothelial cell producing nitric oxide that diffuses and affects the smooth muscle cell. So this is a very clear paracrine mechanism. One cell affecting the neighboring cell. So paracrine regulation of vascular tone. It's important for you to realize that what regulates vascular tone, vascular resistance, vasoconstriction or vasodilation of blood vessels, there are many mechanisms. Nitric oxide is not the only one. Nitric oxide is not the only one. You have the sympathetic nervous system, right? How does the sympathetic nervous system control vasodilation or vasoconstriction? Not saying anything new. I just reminded you not even 20 minutes ago. How is the sympathetic nervous system involved in the regulation of vascular tone, vascular resistance? Acetylcholine, sympathetic. Adrenaline, isn't that more sympathetic? Acetylcholine is parasympathetic. So the sympathetic nervous system, adrenaline. How does adrenaline regulate vasoconstriction or vasodilation? Through the alpha-adrenergic receptors, right? So sympathetic nervous system, noradrenaline. I guess that you are thinking, oh, noradrenaline is released. Well, who releases noradrenaline? It's through the sympathetic nervous system. And that actually is a very important mechanism for regulating vasoconstriction, vasodilation. And then you have paracrine regulation by metabolites, and also nitric oxide. So this is just a small insight. Or I should remind you that vascular tone, vascular resistance, vasodilation, vasoconstriction, the same thing, is actually controlled by multiple mechanisms. Nitric oxide is a very important one, but it's not by far not the only one. There are several. The effect of nitric oxide, more pronounced in large arterioles, while the effect of metabolites is larger in the small arterioles. Remember, this is an important message. There are many points of regulation of vascular resistance in the cardiovascular system. Some blood vessels will be more regulated with one mechanism. Some blood vessels will be regulated with another. It's the total, all these mechanisms that can actually change vascular resistance. In terms of nitric oxide, the main important factor for nitric oxide release is shear stress. Acetylcholine can be important, but it's mainly shear stress on the vascular wall. So, when the blood is actually having or rubbing on the blood cell, that actually will trigger an increase in shear stress. We will see that in a second, the next slide. And that actually leads to nitric oxide. Here you go. This is the picture. This is the endothelial cell layer. Below, the smooth muscle cells, the vascular smooth muscle, VSMC, vascular smooth muscle cells. Above the blood, right? So what do you see here? We know it very clearly that the blood flow in a blood vessel always has a shape like this. That means that the speed in the middle is higher than the speed in the sides. And of course, because of the friction of the red blood cells that are circulating here, you have a friction stress on the wall. Depending on that shear stress, you have mechanisms that will lead to the release of nitric oxide. Which are these mechanisms? Calcium is one of them. What leads to nitric oxide release is the increase of calcium concentration in the cell. That can be through acetylcholine. Acetylcholine, it's GPCR, GQ, PLC activation, IP3, calcium, increase in nitric oxide. Who makes nitric oxide? The endothelial nitric oxide synthase. Nitric oxide diffuses in the vascular smooth muscle cell. What happens there? That targets the soluble guanylyl cyclase that forms cyclic GMP, and actually when cyclic GMP increases in concentration, it blocks the entry of calcium. Calcium is blocked from entry, and when you block calcium entry, basically that leads to vasodilation. Remember that calcium is always the target for contractility, increased contractility. So what cyclic GMP does is blocking calcium entry, and therefore leading to vasodilation. Does this make sense? So important to realize and distinguish here. This is typically a question in the exam, and then what I see is that students confuse the point of generation from the target cell. You should not confuse that. There are different mechanisms. But what's beautiful about this mechanism is that it sort of integrates very well many of the concepts in this course. Notice a GPCR here. Notice cyclic GMP here. Notice paracrine regulation. So understanding this actually means understanding many things that we have discussed in separate parts of this course. Now, we're going to finish before the break with an exciting hypothesis about nitric oxide. A hypothesis that is discussed in different terms. I'm not sure we have a full answer for it yet, but I'm hoping I can actually at least let you to understand what this is all about. The question for this hypothesis is if nitric oxide could have an endocrine role. And I just remind you, what is an endocrine factor? An endocrine factor is that basically has an effect at the distance. An endocrine factor is a hormone, right? A hormone that is released in one part of the body and circulates in the circulation, and it has an effect somewhere else. We have said so far that it works on neighboring cells. I have also told you that nitric oxide has a short half-life, 2 to 30 seconds. Keep this in mind for the discussion that's coming. But this is the fact. We know that nitric oxide can be bound to the hemoglobin. Hemoglobin binds nitric oxide. Remember that that's how actually Luis Ignarro discovered the nature, the chemical nature of EDRF. And this works through a very specific mechanism in which you have a transfer. First of all, the nitric oxide can bind to the heme group of the hemoglobin. The heme group of the hemoglobin in the oxygenated state. Once you have nitric oxide bound to the hemoglobin in the oxygenated state, this nitric oxide can be translocated to a cysteine region in the beta chain, cysteine 93. So you have this that's called heme to thiol transfer. What you're doing is transferring a nitric oxide from the heme region of the hemoglobin to the thiol residue in this cysteine. So you're doing a transfer here. What happens is that we know that hemoglobin, this is oxygenated hemoglobin, but what's the role of hemoglobin? Hemoglobin binds oxygen and transports in the body, right? So what happens when this hemoglobin that is fully loaded with oxygen, at least 3 molecules, reaches a region in the body with low oxygenation? The idea there is that then it will unload the oxygen. That's what happens, right? So basically, this hemoglobin will release the oxygen, and it will come into the deoxy state. What we know, and this is the interesting observation, that in the deoxy state, the thiol groups that bound to nitric oxide will tend to let this go. And this is interesting. Why is this interesting? This is interesting because notice that all of a sudden we are realizing that hemoglobin can transport not only oxygen, but nitric oxide. Why is this also interesting? Because the important thing here is in which condition or where in the body is nitric oxide being released? In an area with high or a low oxygen? Where are we downloading oxygen? Where are we downloading nitric oxide? In areas with high or low oxygen? In areas with low oxygen, which are areas that would benefit of vasodilation. Do you see the point? So the whole concept of this theory is that basically nitric oxide could be an endocrine factor because it can be released at the point where it's actually more beneficial. You will have a vasodilation, and that will expand the vessel, increase blood flow, bring more oxygen. That's a very smart thing. This is a very smart idea. The question is if it holds, if it works. Let me take the second, then we will just switch. What this slide shows is exactly the same thing that I've shown you, but in a different format. So it's showing you the R state of the hemoglobin. This is actually the oxygenated hemoglobin that actually is transferring from the heme group into the thiol group. This is the heme to thiol transfer. And then when this hemoglobin goes through a state of deoxygenation, basically you have a release of nitric oxide. Here you have the nitric oxide. The nitric oxide now can be released, and at the same time that actually the oxygen is given. So this slide shows you the same as the previous slide, it's just different. The question though is, do we have actually support for this theory? This is a beautiful mechanism, isn't it? So the concept is that hemoglobin is not only transporting oxygen, but is also transporting nitric oxide. It's a nice idea. The question is, do we have evidence that this is the case? And I'm going to show you after the break some evidence that suggests, yes, perhaps it works, but some of evidence that is not totally conclusive about this. So this is an area that became very interesting for hemoglobin researchers, and I'll be presenting a little bit of that after the break. But in the meantime, let's take a break. We continue after a quarter past. Don't trample on this cable. I'm actually going to remove it. How I say stop this.
Oh.